So let's start with the first of the basic lecture of biology, that is cell structure. So have you ever thought what living organisms they're composed of? Or rather, what is it that makes us living beings? So if these thoughts have ever crossed your mind, so here comes the introduction to cell. So everything that we're able to do is possible because of the number of cells, that is about 10 trillion cells present in a body. I mean, the number varies, right? Because cells are constantly dying out and they being produced. So the number varies enormously. However, the number is huge. And of course, the function performed by them is even bigger. So let's start with the introduction to cells. If you take a very thin slice of a plant stem and you cut it down and study it under a microscope, we see that the stem, it consists of thousands of tiny box-like structures. And these box-like structures, they're basically called cells, or they're termed as cell. So basically, cell is the smallest unit of life, and most cells, they're so tiny that they cannot be seen with the naked eye. Therefore, we require the use of microscope to study cells. So if we have to define cell, we can define it as the basic functional and structural unit of life. And all living organisms, they're composed of cells. And cells, they're formed by the division of the already existing cells, which in terms of biology means reproduction. And every cell of our body, it comprises of genetic material, which is passed down during the process. So basically, cell is the smallest unit which can live on its own. It can reproduce on its own by the process of cell division. And that is how it increases its number. And moreover, it can pass down that information through generations. And this makes up the living organisms in the tissues of our body. So animals and plants, they're made up of billions of tiny cells. Similarly, like a house, it is made up of bricks. Like any other molecule which is composed of atoms, we say animals and plants or living beings, they're composed up of cells. So the term cell itself, it was introduced by Robert Hooke, an English botanist, in 1665. And he examined thin slices of cork from the bark of a tree using a microscope. He observed closely packed little boxes with thick walls, and these boxes, they look like a honeycomb structure. So this is what Robert Hooke in 1665 observed, that this is how cells look like. However, he only saw the dead cell walls. I mean, you can see the honeycomb structure and the way these cells are. So, however, today our idea of cell is quite different because we can study living cells and with more refined technologies, because now we have a number of microscopes available. So cells are the building blocks of life, the simplest of units which can live independently because they can reproduce independently. And inside, thousands of chemical reactions takes place, and it is these reactions which keep the bodies working, and all these reactions together form the metabolism of a cell. So basically, cells are the simplest of units, building blocks of life, and a number of chemical reactions takes place inside them. That is, they're the site of chemical reactions. And these reactions, they basically allow the metabolism of a cell. So we can imagine cell as literally a chemical factory. I mean, what happens in a factory, that you bring in raw materials for the construction of new molecules. And in cell, the raw materials, they might include sugars, salts, inorganic salts, and these raw materials, they then utilize their process to make new molecules or larger molecules such as proteins, carbohydrates, lipids, DNA, I mean, you name it. So this is basically a thin slice taken from a tip of a plant shoot and photographed through a microscope. And photographs which are taken from a microscope, they're basically called photomicrographs. So this figure, it shows a longitudinal section which passes through two small developing leaves near the tip of the shoot and two larger leaves below them. So you can see the different parts of a stem cells. First, we have the leaf primordia, which is basically a group of cells which will form into new cells. Then we have the apical meristem, which gives rise to the primary plant body, such as here we have the stem part, and the axillary buds, which basically leads to formation of new shoots. So if we have thin slices of this kind, they're called sections. I mean, if we take a whole organism and you cut it into thin slices, we say it's now sections. So if you cut along the length of the structure, we say it's a longitudinal section, such as shown in the figure above. And however, if you cut across a structure that is perpendicular to a structure, we say it is a transverse section. So if you're cutting a cell along its axis or along or parallel to the structure of a cell, we say it's a longitudinal section. And however, if you're cutting a structure across it or perpendicular to its axis, we say it's a transverse section. So the leaves, the buds, the stem, they're all made up of cells. So it is fairly easy to cut sections just by using a razor blade. And to cut sections of animal structures is difficult because they're mostly soft and flexible. So plant cells, they're easy to cut using a simple razor blade. However, with animal structures, they have to be first soaked into melted wax. And when the wax goes solid, it is impossible to cut them into thin sections. And then that wax can be later dissolved away, allowing us to observe the sections of these under a microscope. So sections of animal structures which are examined under the microscope, they're seen to be made up of cells. And however, they're much smaller than plant cells and need to be magnified more. These sections, they're often treated with dyes called stains in order to make the structure inside the cells show up more clearly. So basically, this cells of animal, we're unable to visualize them even under a microscope because they are transparent. So what we do is we use particular stains which basically bonds with these molecules. They make up these intramolecular forces, and that is how these structures, they become visible. So here, for example, here is a photomicrograph of a kidney tissue, which has been magnified about 700 times to show the cells clearly. I mean, you can observe the different structures of cells and we'll go about them one by one. So making sections is not the only way to study cells. What we can do is thin strips of plant tissue, only 1 cell thick, they can be pulled off from stems or leaves. So it's easy to pull off strips from a plant tissue. However, for an animal tissue, what you can do is you can even squash them or smear them on a microscope slide. That is, you spread them on a slide. And then they're treated with chemicals to separate the cells before studying them. So chemical treatment is basically ensures that the cell, its shape is intact, its cell surface and intracellular structures, they remain intact, and the chemicals, they preserve them. So as we said, cell is a unit of life. It consists of mass of living matter, which is termed as protoplasm. Protos, which means first, and plasm, which means form. So first form. And protoplasm is basically a complex jelly-like substance, which is a site for chemical reactions and ensures the life of a cell. So it is a mixture of many compounds arranged orderly. And a protoplasm, it has 70 to 90% water, and the rest remaining, it consists of mineral salts and organic compounds. So protoplasm is basically the living essence of a cell, and it is composed of, most of the protoplasm is watery, and the rest is of mineral salts and organic compounds. So since protoplasm is a mixture, its composition, it varies from plant to plant and animal to animal. And similarly, if we're talking about organisms, that protoplasm is varying between organisms, we say that it varies even between the different types of cells, such as the protoplasm of a muscle cell, it is different from that of a brain cell. So protoplasm exists in 2 forms. One is the solid state, which is salt state, which is the liquid state, and the other is the gel state, which is the semi-solid state. So basically, we say that these 2 states, it depends on the physiological nature of a cell. And protoplasm, it consists of three parts, mainly three parts: the plasma membrane, which is otherwise known as the cell surface membrane, the nucleus, and the cytoplasm. When it comes to studying animals or plant cells, we say, or we biologically believe, that there is no such thing as a typical plant or animal cell. And that is because cells, they vary a great deal in their size and shape depending on their function. However, we have certain features which are present in most cells, and that is a cell surface membrane, which is basically a thin boundary enclosing the cytoplasm, and a nucleus. So these three structures, a cell surface membrane, cytoplasm, and a nucleus, they're basically the common features present in most cells. So this is a section through a liver cell of an animal, and this cell has all the characteristics of an animal cell. For example, you can see that we have a cell membrane, which is a thin boundary of the cell making up the boundary of a cell, and its function is to control what enters and leaves the cell. That is, that the molecules which enter and leave the cell, it's basically acting as a barrier. Then we have the fluid part, which is the cytoplasm. Then we have the fluid part, which is a cytoplasm, and basically metabolic chemical reactions occur here. It is a site of chemical reactions. And another enclosed membrane structure here is the nucleus, which basically contains DNA and so controls the cell. We'll cover the function of DNA later. And within the nucleus, within this membrane-bound organelle, or within this membrane-bound structure, we have DNA, which contains the coded instructions to make proteins. We talked about how cells, they can reproduce independently, they can survive independently. So these structures, and basically DNA, which contains instructions for a cell, this allows cells to replicate independently. That is, it allows a cell to divide or pass on its genetic information to the next generation. So we saw how a typical animal cell might look like. So this is how a typical plant cell looks like. I mean, you can compare and contrast the different features which are present in plant cells and absent in animal cell. So we have a cell wall, which is cellulose. It is composed of cellulose. And following the cell wall, we have a cell membrane. So basically, cell wall is the protective barrier, or another outline, you can say, to a plant cell. And like a plasma membrane, which we say, is semi-permeable, that is, it acts as a barrier to the entry or exit of substances. Cell wall, it does not act as a barrier, or it does not affect the movement of ions or substances into or out of a cell. And then we have a large central vacuole. And within the cytoplasm of a plant cell, we have many different membrane-bound structures, such as mitochondria, chloroplast, ribosomes, rough endoplasmic reticulum, smooth endoplasmic reticulum, amyloplasts, and a plant cell also contain a nucleus. So you can see that cell membrane, cytoplasm, and nucleus, the three structures which basically combine together to form a protoplasm of a cell, they're present in both animal and plant cells. So cytoplasm is part of a protoplasm surrounding the nucleus. And under a light microscope, the cytoplasm, it looks like a thick liquid with particles in it. In plant cells, it may be seen to be flowing about. So it's basically a jelly-like structure, and it is a site of many chemical reactions which keeps the cell alive by providing energy and making substances. The particles, they may be food reserves such as oil droplets or granules of starch. Other than that, it also contains structures which we call them as organelles, each having a particular function. So an organelle is basically a highly organized structure found within a cell, that is a subcellular structure which has specific function. And these organelles, they might be mitochondria, which is basically small spherical rod-like organelles, and they're involved in release of energy from food substances during cell respiration. And energy release may be temporarily stored in small molecules called ADP, adenosine triphosphates. So mitochondria are basically the energy producing organelles found in plant cells and animal cells. And that energy, it is stored in the form of ATP. So ATP, they are the batteries in a cell, and they can be transported from one part of the cell to another. When needed, they're broken down to release energy. This whole mechanism will be covered later. And energy energy released by the breakdown of ATP, it is used by the cell for growth and reproduction. So, and since ATP is a small molecule, it can be transported from one part of the cell to another. So whichever part needs energy, ATP is delivered there and then broken down to release energy for growth or reproduction. So the liquid part of cytoplasm is about 90% water, with molecules of salts and sugars dissolved. And suspended in this solution, there are larger molecules of fat, lipids, and proteins. So lipids and proteins, they may be used to build up the cell structure, such as the membranes. So lipids and proteins, so when we cover the chapter structure of membranes, you'll see how lipids and proteins, they help in the structure of a membrane. Some of these proteins, they're enzymes, and some enzymes, they're attached to the membrane system of the cell, whereas others float freely. So a large part of cytoplasm is basically water, which has all these substances which are required by a cell to survive, which include salts, sugars, lipids, and proteins. So cell membrane, which is basically a thin layer of cytoplasm around the outside of a cell, it's basically enclosing the whole cell. It stops the cell contents from escaping and also controls the entry and exit of substances. In general, oxygen, food, and water, they're allowed to enter, whereas waste products, they're allowed to leave. And moreover, harmful substances which can harm the cell, they're kept out. So in this way, the cell membrane maintains the structure and chemical reactions of the cytoplasm. So basically, since it's enclosing the whole of the cell, so it's helping in maintaining the structure of a cell and moreover chemical reactions by allowing the certain molecules such as oxygen, food, and water to enter, whereas other molecules, that is waste products, they're allowed to leave. So the third common feature, the third common structure, nucleus, which is a small spherical mass of denser protoplasm, we say the nucleoplasm. So a rounded structure enclosed in a membrane. The nucleus itself is an organelle. It is enclosed in a membrane, which we say is the nuclear envelope. And it is embedded in the cytoplasm and controls cellular activity. Most cells, they contain only one nucleus. And nucleus, it may be shown darker than the cytoplasm because it takes up certain stains more strongly. So whenever we visualize a stained cell under a microscope, the darker region is usually the nucleus. And its function is to control the type and quantity of enzymes produced by the cytoplasm and also able to regulate the chemical changes which take place in the cell. So as a result, it determines the fate of a cell, that whether a cell will become a blood cell, a liver cell, or what kind of cell it will be, what kind of a cell will be. However, within the nucleus, we have following structures. That is a network of long thread-like structures called chromatin, which contains hereditary material, that is material inherited from parents. So a thread-like structure, chromatin, is basically present within the nucleus and which contains important information which is being passed on from your parents to you. And during cell division, the chromatin, it condenses and becomes coiled, and we say it has become chromosome. And these chromosomes, they appear as thick rod-like shape under a microscope. So basically, these chromatin, during cell division, they become condensed and coiled to form chromosomes. Spherical structures called nucleoli, which plays a part in building of the proteins, and the nuclear envelope, which separates nuclear content from the surrounding cytoplasm. So in a non-dividing cell, DNA is present in the nucleus in the form of chromatin. And it is only when a cell is dividing that DNA, it becomes condensed and coiled to form a chromosome. So each chromatin thread is basically composed of proteins and DNA, and it is in DNA that the hereditary information is stored. The nucleus also contains cell division. That is, a cell without nucleus, it cannot reproduce or replicate. So inside the nucleus, there are thread-like structures called chromosomes, and which are easily seen at the time when the cell is dividing, because the chromosomes, they condense, they coiled up, so they easily easy to visualize. And we have been saying that cell that is able to replicate independently. So this is a diagram, a graphical representation where an animal cell it is about to divide. So we can observe here that the nucleus, this is the nucleus, the denser part, and the cell membrane and the cytoplasm. So we can see that in this diagram, nucleus divides first, and then cell divides or the cytoplasm divides, such as the daughter nuclei, they separate, and the cytoplasm pinches off between the nuclei. And here we see that the two new cells are formed, and one may keep the ability to divide, and the other it may become specialized to perform a particular function. So however, in cell division, you have to remember that the nucleus, it divides first, and then the cytoplasm pinches off and the cells divide. So here is the structure of a plant cell. You can easily visualize that it consists of a cell wall, an outer structure, which is the outer line. So plant cells are basically composed of two outliners basically. The one is a cell wall and the other is of a plasma membrane or the nuclear membrane, which is enclosing the cytoplasm. These green structures, these are the chloroplast, which is basically a site of photosynthesis. This orange sapphire is basically the nucleus, the denser part. And in between, we have a large central vacuole. And the yellow liquidy jelly-like structure observed here, it is basically the cytoplasm. So let's end the lecture here, demo lecture here. The time is about to run out.